Let's take a look at the concept of race, and we want to do so from an anthropological, a biological perspective. For example, according to the American Association of Physical Anthropologists, they revised the UNESCO Statement of Race in 1964. We want to talk a little bit about that, and we're going to review our understanding of genetics, and that's going to help us develop an appreciation for this concept of race, which ultimately is a folk taxonomy that has no basis in biology. Let's move to the next slide. So, looking at the concept of race anthropologically, as pointed out, in 1964, the American Association of Physical Anthropologists revised the UNESCO Statement on Race. And what they wanted to point out was that all extant or living human beings belong to a single species and share common descent. They also go on to argue that variation it does exist between humans. However, this has been shaped by hereditary, environmental, nutritional, and cultural factors. And in the end, there is no basis for the idea of race in biology. As pointed out, all extant humans belong to a single species. We share common descent, and this is something that Charles Darwin had pointed out as well. And reflecting back on Charles Darwin, he also said or stressed the importance of variation. And while some individuals in the United States have gone after Charles Darwin trying to illustrate that he was a racist, that really didn't figure into his origin of species. Charles Darwin recognized that variation was essential to the success of any species, including the human species. So here, as pointed out, variation, it's been shaped by environmental forces, it's been shaped by nutritional forces, and cultural factors as well. And furthermore, we also want to say that there's no national, religious, linguistic, or cultural or economic class that constitutes a race either. Nevertheless, people continue to use the race, and the worrisome thing about this is it's insidious at the level or underhanded in a way that it divides human beings into subgroups that continue to experience differential treatment on the basis of perceived physical features. Let's move on to the next slide. To develop an appreciation for the differences that exist between different populations, we have to take a look at DNA and our genes. DNA, was discovered by Watson and Crick. They were the first individuals to create that model, that double helix model of DNA. And what we know about it is a complex molecule, and it's made up of four basic bases. Now, here are four bases. We see them depicted on that individual. It's the arrangement of those bases that are responsible for everything from the tone of our skin pigment to the color of our eyes to our ability to fight infections and on and on and on. So here we have our four bases. For example, adenine, we have our guanine, represented by G, we have our cytosine, represented by C, and of course our thymine, which is represented by T. And as pointed out, it's that arrangement that we find on that double helix. And if you're wondering where that exists, I'm going to try and sketch it in over here. So we have our double helix, and it's almost on this ladder, these rungs, for example, that we find that arrangement of the A's, the C's, the T's, and the G's. And then a little bit higher up, we might have C, T, G, A. And it's that arrangement that eventually becomes unique for populations that creates the differences that we see between us. Let's move to the next slide. Now, how do these differences emerge? Ultimately, what we're looking at is something that's referred to as a copying error, a mutation. It's a random event that takes place that causes a rearrangement of those base pairs on that strand of DNA. So, put simply, sometimes DNA doesn't copy itself accurately. And as pointed out, this is a random event. And if you think back to biology, This process, we call it mutation. And that's the only way that new genetic material gets into the human species or any population for that matter. So new features, they will only emerge through this type of copying error, this mutation that takes place, and that produces differences. So these mistakes, they can lead to new arrangement of base pairs, and in the end, the arrangement, if it's beneficial, if it provides the species, the individual, or the group of people with an adaptive advantage, as Charles Darwin pointed out, it's going to be or can be positively selected. So over time, unique features among populations can emerge. Let's move to the next slide. So we have our DNA and we have our 4 base pairs. And what we want to consider is on that strand of DNA, we have a particular segment and we're going to refer to it as a gene. Now, it's the order of these bases in those particular loci or those genes that determine the information that's available for building and maintaining an organism. And as pointed out, this ranges from all of our physical features, for example, our skin pigment, our eye color, the type of hair that we have or the absence thereof, our blood type, our ability to fight infection, and on and on. All of that is determined by this arrangement of base pairs. It's a lot to process, but I'm going to try and make this perhaps a little easier. Let's think of words in the alphabet. So the alphabet, it's really in itself, it's just a number of symbols that we've attributed meaning to. And on their own, we have B, and we're just going to say it's a letter. But if we begin to add and combine letters together, we're going to produce words. And if we begin to take those words and we combine them, we're going to get sentences. So overall, we do get this meaningful system beginning to emerge by simply putting together and rearranging letters. So instead of looking at A, C, T, and G, I'm going to use something here as a replacement, D, E, A, and M. Now, if we rearrange those words, those letters, sorry, we're going to get, for example, made, M-A-D-E. Now, looking at those letters, we can rearrange them again. Now, let's say we apply the principle of biology here, and through a random mutation, we get a rearrangement of made, and it produces dame. Again, another meaningful word to us. We can imagine this as a new type of physical feature. And yet again, we can take those 4 letters and we could rearrange them yet another time and get a tasty cheese, for example, Edam. Like I said, this is the way that evolution works. This is the way that new features work. And this is how we begin to explain the differences that exist between populations. So, for example, made, let's take a look at that. And let's say that's the original arrangement producing skin color. And as long as humans, Homo sapiens sapiens, have lived on the earth about 200,000 years, our species or our ancestors have always been dark-skinned. That was the first human dark-skinned individuals. But eventually, there was a mutation, and it produces the D-A-M-E. Light skin emerges. Now that provided some individuals with an adaptive advantage in cooler environments. I'll talk a little bit more about this. And that new feature, it was selected for. So this is the way that evolution and variation works. Let's move to the next slide. Now, I've referred to skin pigment, and this is usually a question that a lot of introductory anthropology students, they ask me. They say, you know, I don't mean to sound racist. I said, well, be careful. And they said, okay, I'll be careful. You know, what was the first color of humans that existed? I said, well, that's not a racist statement. We're not saying anything bad about individuals. This is a legitimate evolutionary biological question. And as pointed out, the simple answer is Homo sapiens sapiens, our ancestors, had dark skin first. That doesn't mean that we didn't see variation emerge over time. Now, how did that variation emerge? It emerges through something that we call a single nucleotide polymorphism. We call it a SNP for short. And here, for example, we see SNP and an A, G, and T just in that segment. And what we have is a reversal of those base pairs that we've already talked about. Now, the oldest skin pigment, as pointed out, was darker pigment. We're not sure how dark, probably an intermediate tone. Darker skin, that seems to be more of a more recent adaptation. But the gene that's responsible for selecting for skin pigment is referred to as the SLC24A5 gene. And here we're looking at the variation A11T. Now, the original gene here that's selected for skin pigment, light or dark skin, was SLC24A5T11A. All that happened was a shift, a rearrangement between that T and A. They changed position, a 111 position on that gene, and that took place around 12,000 years ago, and that leads to the emergence of light pigment. Now, like I said, for these types of features to remain within a population, they do need to have some sort of selective pressure. Now, the pressure for light skin and the adaptive advantage of that was in cooler climates where vitamin D, or we should say that UV radiation, was much lower, and it was much more difficult to absorb vitamin D when one's skin pigment was darker. So individuals that had this mutation, they were able to get more vitamin D. Their bones developed properly over time, and that was an adaptive advantage, and it stuck. So interestingly, it wasn't until about 6,000 years ago that we start to get a broad sweep of individuals with lighter skin. Let's move to the next slide. Let's sum up and draw a few conclusions here. So we've talked about DNA. We see the DNA spinning here. We'll go ahead and label it. Hopefully you can see it on your end. There's DNA, and it's composed of a number of different base pairs off to the side, A, C, T, and G, and is the arrangement of those base pairs that's responsible for creating differences in us. Now, as for our physical features, and here we'll just go to the center of this particular DNA strand, and we have a segment or a loci, and we refer to that as a gene. And it's that gene that's responsible for producing different types of physical features, or what we refer to as our phenotype. So here we'll say our physical features. They're produced by our genes. And when we see differences in physical features, it's a result of a variant gene. So it's on that same position, but something has changed there in that A, C, T, and G, and it produces what we call an allele. So human biology and race, it's intertwined. It's the only way that we can understand whether or not races in fact exist. And quite frankly, there just isn't enough diversity between the populations. Even though we look at different populations or ancestries, and we tend to think, wow, we're different. This is often because we've been trained to think in basic dichotomies. We've been trained to think as people with darker skin as black, and we've been trained to think about people with lighter skin as white. And you cannot get any further apart than being black or white. In contrast, Angelica Doss, she has a project referred to as Humanae, and she illustrates that in fact there is no tone in the human species that is in fact black, and there is no tone that is white. In contrast, we have a spectrum, a range, like a range of light, and we all fit into that spectrum one way or another. And our differences, we don't mean to undermine them. We don't want to say that. We want to say that our differences are important. This has created the faces of success when we look at humankind. So when we look at our genes, there's some 25,000 genes in the human body. And looking at those differences and calculating how different we are, for example, people of African ancestry versus people of European versus people of Asian ancestry, we're 99.7% genetically identical. And that is not enough to create what we would define as a separate biological race. We've pointed out that the variation that we see, this occurs in these single nucleotide polymorphisms, and the difference is it's 0.3% among populations. That's the difference that exists between us. We've talked a little bit, as an example, we've looked at the SLC24A5 gene that's responsible for selecting pigment. And we had pointed out that the earliest skin pigment that humans had was dark pigment, and there was a mutation along the way where that T111A position, those two base pairs, they reversed position, and we have SLC24A5 A111T, and that was responsible for producing lighter skin. Now, this is important to understand because it shouldn't separate us. What it should do is illustrate the significance of biological evolution. And even though we diverge in different ways, we still remain a single species. And as pointed out, when we look at our faces, we look at our differences. We shouldn't see the strong differences that exist in an effort to separate us. But we should be looking at ourselves in a way that we say, wow, we are a successful species. We've diverged in a variety of different ways, and we continue to maintain an interbreeding population. We are not a subspecies. We haven't turned into different species like other animals do. We don't have different races. Let's move on to the next slide.